Good morning. Hope it's still good morning, yeah. And I'm not going to change it into the negative side. Yeah, so it's me. So it's actually the name of the company. We are still not allowed to go with this name externally. So we are still Janssen Pharmaceutical Company of Johnson & Johnson. Also internally, we already changed the name into the J&J Innovative Medicine, but it's still internally. So it means I'm Janssen. And yeah, so we are a pharmaceutical company. So the products what we develop actually need to find the proper position. So we actually develop in drug therapy, medicine. So, but I'm going to talk today about the endpoints and biomarkers in a clinical trial. So this is actually something what we are using, or I would better say that we are actually hope to use maybe in the future, because it's not, the field is not much mature yet. We are already doing a lot, but still not yet there. So this means it's not our product, what we want to advertise, but what we are doing and how it's actually all this stuff like a digital endpoints and biomarkers could be developed. So this is a master class, and I will try just to give some kind of guidance about the different possibility, different solutions, and different direction in this field. Just to start, I probably go through the definition. And I will go through the one very general definition of what is actually something digital. Why I just want to start from this definition of digital, because it's a lot of time it happens that people are coming to us and they're trying to advertise quite a lot of products, saying that this is in the field of the digital health, but in reality, just simply standard digital products, yeah? Maybe some kind of database, maybe some kind of website, and so on. But since they feel that it's necessary for a pharmaceutical industry, they also try to put it into the field of digital health, which is not true. And I'm going mainly just to concentrate more into the topic of the digital health, which is still quite broad. Digital health can include quite a lot of things which is not necessarily be useful for directly into the clinical trial, but it could be useful, for example, for, I don't know, for the doctor, for the GPs, like some kind of tools for the telemedicine. So this also falls into the digital health. So digital health, we can actually, let's say, put some kind of other stuff which is more general versus digital medicine. Because maybe just first come to the digital medicine, because here I want just to highlight this is evidence-based technology used for measurement in intervention during the clinical trial or maybe outside of the clinical trial. Digital health is a more bigger one. Like for example, if you're talking about the digital health, it could be, say even the system just to record the data. Yeah, so something just to do the decentralized clinical. Not necessarily something for the measurement or directly like a measure or intervention. It could be maybe system just to track where the patient actually taking the drug, yeah? Some kind of smart blisters based on which we can see the patient is actually taking drug or not. Or maybe it could be some kind of system like, I don't know, video monitoring through the smartphone where the patient actually taking the drug or not, where the patient is swallowing the drug or not. So it's more general what we call like a digital health. But the digital medicine, it's more exactly evidence-based, where there is some kind of evidence was generated, and this exactly about the new measures is some kind of intervention. When we're talking about the digital medicine, so the primary people are putting these 4 different categories: digital therapeutics, digital phenotyping, digital endpoint, and biomarkers. When we talk about the therapeutics, it could be, for example, some kind of particular games which help with some kind of disease. Like for example, there are already several games developed for autism, when people are playing this game and based on this, okay, so their measurements, there's some kind of criteria how the autism is measured, it's actually improved. Or maybe it's some kind of a companion apps on a smartphone, and the people are taking some kind of drug, but drug probably produce some kind of side effects, and those accompanying apps based on which people can, I don't know, try to ask the question, get an answer how to deal with all these side effects. Here I will primarily concentrate more, and let me move to the next slide, onto this part, digital phenotyping, more precisely digital biomarker and digital endpoint. When we're talking about the phenotyping, this all around the measurements of what is happening to the people. So whether there is particular correlates of the disease. And this primary and a lot of used in the academic research. I believe majority of the papers what we see published in this field that exactly could fall into the digital phenotyping category. But we are in digital phenotyping, it's not necessarily regulated by the FDA on EMA. But when we are coming to the biomarker, and more importantly to the digital endpoint, so regulatory, it's played quite a huge role here. When we are talking about the biomarkers, so I listed the several types of the biomarkers, and those are used, for example, for stratification of the patient, maybe used for prediction of the outcome of the clinical trial with a specific group, or maybe our prediction of the decline or maybe improvement of the patients. And when we are talking about the endpoint, this is something that is used in the clinical trial. And here when we are talking about the endpoint, I would probably divide this into the two parts. One part, what I will call clinical outcome assessment, and second part is like a surrogate biomarkers. So what is the difference? When we are talking about the clinical outcome assessment and something what is used in the clinical trial, based on which we can say that our medicine works or not. But this is something which is characterize something meaningful for the patient. While when we are talking about the surrogate biomarkers, which also you could be used like an endpoints, here it's not necessarily be understandable for participants, for patients, but still could be used to track how the medicine works because it's related to something which is already meaningful for the patient. Probably what I just mentioned is quite, I don't know, quite general, so we'll come to specific examples later on. Well, probably all this stuff will be a little bit more better. But before we'll come to this, I'm not sure, so this is not my company, but I believe this news, which is appeared when it was, I believe it was in September, yeah, maybe 2 months ago. So it was quite, I don't know, a big thing for the pharmaceutical industry and especially for the development of the digital endpoints and biomarkers, yeah. And this news said that Biogen, because of the situation where they are right now, and yeah, we know right now we're in the recession everywhere, and they just simply decided to close their digital health group. And they decided to lay off about 150 people. So in the digital health world, so this looks like probably already there is something changing in the industry, how we are viewing all this stuff. Because previously people thought that, okay, so it's, yeah, so it's the future, we need to invest in these particular things. But with what's happened with Biogen, so people start to rethink. And first, okay, so when people see, they already react to some kind of negative news. This is something negative and so on, and usually people do not read also all the news carefully. And what I want to show, so this was some kind of additional insight about what's actually happened in the Biogen, yeah? So this is a post from the co-founder of the Human First, from Andy, who, let's say, share exactly how she feels and what exactly happened in the Biogen. And this is interesting to read, because you see, so it says that the Biogen decided just to go away maybe right now from the different symptom tracking tools, yeah, which could be in the field of the digital phenotyping, yeah. They go away from the companion apps on digital therapeutic, right? They so stop also, let's say, some kind of observational trial with Apple. They move away more from the decentralized clinical trial, but at the same time, they keep and put a little more attention to the digital biomarkers, rare diseases, imaging tools. So why I'm showing this stuff? Because as you see, even from Biogen and from our perspective, what is more important, it's exactly just to concentrate into the biomarker and the endpoint, how to help in our clinical trials. So it's not simply monitoring of the participants, not simply understanding the disease, but where that could be served and something to help us in our clinical trial development, our development of the drug. Before we'll go further, I just want to introduce one thing which I believe quite important from regulatory standpoint. So this is so-called concept of interest. So if you have a particular disease exhibit, I don't know, and the different symptoms and the different things, Yeah, so what is listed here. And probably all this relate to some particular bigger, broader concept. And this concept, like for example here, lower limb related function, could be split at more simple, smaller concept, like walking capacity, like muscle strength, and so on. And those concept of interest, it's exactly what could be measured with the different devices. So this means always, let's say, when you even submit something to the regulatory authority, you first need to mention it was a concept of interest for your development, for your drug development tool, and next already about the technology. Let us go through some kind of example. So first example, it's 6-minute walking test, which is quite broadly used. Like for example, it's used in the pulmonary hypertension. So what's the idea of this? So there is a lengthy corridor, so there is 2 cones, yeah, and the participant is asked just to walk around across those, so from one side to another, and walk for 6 minutes, and the distance which the participant covers, so it's actually used like a measure in clinical trial. So idea is, if you have a drug, if drug works, and drugs touch at exactly exercise capacity, which is concept of interest, in this case, we hope that through when patients are taking this drug, this particular medicine, so the distance what they cover will improve, because we think that this should correlate to this exercise capacity. It's not simply that participants need to walk, just, I don't know, very relaxed and so on. The idea is that person need to move to the topmost of their capacity. So this is why it's usually done in the clinic. When the clinician saying, please go faster, go more, and so on and so on. What is the problem with this stuff? So first of all, as I mentioned, so we need to encourage participants just to move as fast as possible, as more as possible. It was already shown that if you promise that we are going to give you, say, $100, 100 euros, the participant will cover higher distance. So this means we know that, okay, so from time to time, depending on the motivation, so the distance coverage will be completely different. Another problem is that actually in pulmonary hypertension, so if you look into the participant, how they are moving at home, they are moving in street and so on, they usually not go more than for 3 minutes. So this means 6 minutes, it's already quite a lot for them. That's not what they naturally do. So this means this test is completely not natural for them. So this means what we are using in a clinical trial like this, it's not really great, and maybe we can do something better using some kind of, I don't know, digital technology. Another example, it's epilepsy. So how we are running right now clinical trial when we are developing drugs for epilepsy. Usually, how it's done, so there is some particular either paper version or some kind of electronic version. So I just took something from the internet, so not necessarily this one, right? So, but mainly, there is some kind of like a diary where a participant is asked to keep track of how many seizures they recall happened during the last day or maybe last week. Problem is that everybody knows that our memory does not serve us well. Also, there is some kind of seizures called like absent seizures where people simply don't notice that seizure happened. So if seizure happened during the night, I'm not sure that you will recall that the seizure happened. Probably you will don't notice. So everybody knows that this recollection and so on is not a great stuff when you say how many seizures. But nevertheless, it's exactly what is used right now in our clinical trial. So here I'm just simply showing graph. When it was done some study in the hospital, when from one side people were video EEG monitored, which is used like a golden standard for the epilepsy, and they also were asked just to keep a diary and say how many seizures happen. And you see there is no really great accordance with this. So here, a concept of interest is epileptic seizures, and that's what is used right now, but again, maybe we can use something better and develop something better using some kind of digital technology. And both of those, like in the previous slide, the 6-minute walking test, and this seizure counting, so this is an endpoint in our clinical trials, based on which we can say whether the drug works or not. Right now, coming to the development of these digital endpoints. Those two examples, what I showed in terms how they going to be developed, this new digital endpoint, they actually follow 2 different paths. First path may be looking here, and there's a 6-minute walking test. As I mentioned, 6-minute walking test and something which measure exercise capacity. Exercise capacity is some kind of concept what we have in mind, but I'm not sure that we can directly measure this concept. It's like intelligence also, yeah? Intelligence, we cannot measure it directly, so that's why we have a different test, like IQ test, I don't know, some schools exams and so on, yeah? So it means there are a lot of different tests which measure some kind of hidden or latent concept. So here also, let's say we have this exercise capacity, there is 6-minute walking distance test, there is some kind of other tests, and if you're developing something new, we're developing something which needs to measure some kind of hidden stuff. Second thing is, we're like with epilepsy. With epilepsy, we actually can measure this ground truth. As I mentioned already in the previous slide, like a video EEG monitoring can give us exactly what is happening here. So this means what we have, we just measure and want to have some kind of additional more, I don't know, because you cannot do video EEG monitoring at home all the time. People need to be in the hospital. And in this case, clinical trial will be quite expensive. You want to do something and measure at home of the people, probably you want to use some kind of digital device, but this digital device needs to be approximate, or will be, let's say, I don't know, coincide completely what we have from the video EEG monitoring. So these two scenarios, so they actually require a different way of the development endpoints and what kind of things regulatory people are actually asking for this. Let us come to this first scenario when we are measuring something which is hidden, yeah? We cannot say, let's say, that we want to correlate to something here because we don't know what is there, right? So it means we need to develop something and need to develop something first to show that this, what we are developing, it's quite relevant to this, yeah? What I mean here, this is called validity. Validity is that we exactly measure this hidden variable. How to show this? We need to show that there is relation with some kind of other measures which already were used, yeah? What I mean, for example, about this exercise capacity, this means whether this new measure relate to the 6-minute walking test, relate to something else, and so on. But this relation needs to be high, but not really high. Because it will be really high, so it means it's already, let's say, quite nicely mimic 6-minute walking test, which we already showed the bad stuff, right? So it needs to be close just to show that we are measuring the same, but not really close. Next, what we also need to show, it's reliability. What means reliability? People primarily know test-retest reliability, yeah? So if nothing changed to the participant, if you do it twice, we need to get the same result. As I mentioned with the 6-minute walking test, if person was asked to perform a 6-minute walking test twice, we need to get the same result. If it will be not the same as I mentioned, we promised 100 euros and the distance was higher, it's not really great test-to-test variability. Yeah? So it means we cannot rely much on this measure. And for sure, like for all measures, we also need to show that this measure is actually sensitive to change. What I mean sensitive to change, if there is improvement or maybe deterioration in the health state, this measure has to capture this. So this is our first scenario. Second scenario, when we have a ground truth, in this case, we need to also perform validity and sensitivity to change, but we don't need to do reliability. Because reliability is not necessary, because if there is already a relation to the ground truth, and this is a good relation. In this case, already our measure more or less stable. How it's going to look like for development of the exercise capacity? Assume that we have a variable device, and variable device provide us, say, step counts every minute. That's what I'm showing here. A lot of people actually use it, and this was used like a primary endpoint in one of the clinical trials, the phase 3 clinical trial. People primarily use into the time people spend, let's say, in the sedentary activity, time people spend in the moderate activity. It was used, but it's actually failed. And here probably there is another possibility just to look at this. So if you look for a long period of time and just count, let's say, minutes, not look into the minutes when there is a steps. So like, for example, in this minute we have 21 steps, 28 steps, and so on. And next aggregate all this stuff and construct a histogram. Construct a histogram and, for example, take 95 percentile here. And just to use the measures which correspond to the 95 percentile. Why we are doing this? Because all of us are walking sometimes like a recreational walk, sometimes we are walking fast. But as I mentioned, if we are coming to the exercise capacity, we need to be in the top of our performance. So that's why we want to be here to the right as much as possible. But not completely to the right, because definitely all the time will be outliers. If you use such a measure, we can show that this measure is quite valid. I'm just here showing only the relation to the 6-minute walking test. And as I mentioned, this validity needs to be high, but not really high, because we don't want to have something which is exactly like 6-minute walking test. So this means this correlation, what we are getting about maybe like a Spearman correlation 0.5, it's already considered quite nice. It could be also shown that the same relation is for other, maybe questionnaires and so on, that we measure the same stuff. Next, we can also look into the test-retest reliability. As I mentioned, so if you do it once, doing it second time, it needs to be more or less the same measure. Otherwise, the measure will be not much reliable. And this is exactly what's shown, that this measure is quite reliable. So this means we already see that this measure can cover two things, like it's a valid measure of exercise capacity, and it's also a quite reliable measure. I'm not going to show results about sensitivity to change here, because, okay, so in this case, it will be quite several other slides. But you can trust me that also the sensitivity to change here, it's also really nice. Here, what I wanted to show, that for this particular measure for its development, we already showed that measure it's reliable, and measure it's valid measure of exercise capacity. Moving to our second example, about the seizure event, seizure detection. So here we can maybe first let us think a little bit about the epilepsy, about the seizures. We already know quite a lot about the seizures. We know that when seizure happens, in a lot of cases, yeah, so there is some kind of motions which has happened. Yeah, probably people saw people with epilepsy and they saw when there is a seizure, so there is some kind of shaking events, right? If there are shaking events, probably we can use accelerometer or some kind of motion tracker just to grasp exactly the time when the seizure happened, right? So here in the graphs, it's exactly, let's say, the accelerometer, 3D accelerometer, and you see when there is a seizure, you have quite a lot of different oscillations, yeah? It's a good indicator, but the problem is, I believe, if you are in your morning, in the evening, brushing your teeth, you will probably have the same behavior. So if you look into your data during the brushing your teeth, it will look more or less the same. So this means, although actigraphy measures acceleration, it's quite nice, but only using this measure will be probably not possible just to discriminate exactly when seizure happens. We know additional things about the seizures. And a lot of time, even before the seizure starts, maybe for 1 minute before the seizure, always the heart rate is going up. So you have a tachycardia. So this means if your sensor would be with your sensor would be possible just to detect your heart rate and just notice that heart rate is going up, probably it also will be some kind of characteristic of the seizure. But again, heart rate is going up during a lot of other events, right? You're doing sport and heart rate is going up, right? We know additional things about the seizure. We know that in majority of cases, after the seizure, there is actually some kind of, you start to sweat a little bit. So what could be recorded and identified based on electrodermal activity. That's what is shown here, for example. So this is seizure event, and you see, so the electrodermal activity is going up after the seizure. So because you are sweating a little bit. But again, the problem is that you sweat also after the sport. Maybe you're going to the sauna and you're sweating, yeah? All of those readouts per se would be good to tell you that there is a seizure. But maybe taking all this stuff together, in this case, you can develop some kind of nice seizure detector. So for sure, so here it will be for each participant, so how all this stuff look like will be different from seizure to seizure, it will look like different, but still there is a possibility. Maybe it will be not possible to develop this seizure detector for all type of seizures, yeah, like partial seizures and so on. But some of the type of seizures, like generalized tonic-clonic seizures and so on, so it would be possible to do. So I'm here showing like an example what was done in one of the consortiums, this IMI Innovative Medicine Initiative, several years ago. So they already developed, and yeah, probably I'm not going to go into the result, but mainly I wanted to show that This, because we have a video EEG monitoring, it's exactly what's in the clinic. So when the people were video EEG monitored, like a ground truth, and the device which was worn, yeah, so it was possible to develop an algorithm which would be able to detect the seizures, more or less precisely, for sure, with some kind of false positives and so on, but still it would be much more better than just ask people just to keep a diary. And maybe just to wrap up, in the pharma industry, I believe what I show from the Biogen example, and also what we experience in doing in the Janssen, we are primarily interested in the digital biomarkers and endpoints, something which could be used for the clinical trials, based on which we can identify that our drug works or does not work, right? Or maybe could helpful for select a population to the clinical trial and predict who will be the people who are going to benefit from the drug. As I showed with two examples, depending whether we have a ground truth data or don't have a ground truth data, there is slightly different approach. Like, for example, whether we need to show reliability or not. So this means it's quite important if you're developing new endpoints or biomarkers, always be in touch with the regulatory authorities, with FDA and EMA, ask their opinion from the beginning, and just to follow all the recommendations. So if I would say that regulation is not really greatly nicely described, so this means if you're doing something, you need to do it, let's say, and ask all the time. And for sure, let's say digital biomarkers and endpoint is quite important. It's help just to increase trial success because they are more objective, what I showed. Yeah, we are not relying on the human memory just to recall how many seizures you have, but we are measuring more objectively. So this can help, let's say, just differentiate product from competitors. What I mean, like, for example, based on these new measures, we can show some kind of additional stuff which is not possible to measure with other things, other endpoints, and this probably could be some kind of additional things what we can put on the label, the segmentation of the label. And for sure, conduct trial faster, better, maybe reduce population which is needed into the clinical trial. Thank you. Would be happy to answer questions.
Thank you very much. We do have some time for some questions. Please raise your hand.
Hello, thank you very much for the presentation. Sebastian Schwartz from Oken. I was wondering if you could comment a bit on the dynamics of actually deploying such a biomarker, a digital biomarker, digital endpoint in a new trial. Like what happens within a pharma to actually say, okay, we're going to use this new digital biomarker, and how does this happen, both internally and also with regulators, yeah, to make it work.
Mm-hmm. So, as I mentioned from the beginning, I believe right now we are still more in the development stage. Yeah. So if you are talking about the inclusion into the clinical trial and making some kind of claim based on this, there is actually two approaches. All needs to go through the regulatory space. One approach is just to classify your endpoint like a drug development tool. And this was done, I believe, only once. Was done not in the United States, but this was done in Europe. It was done for the Duchenne disease. And it was about 95 percentile of the stride velocity. So this means there is just only one approved endpoint in Europe to be like an endpoint in a clinical trial. Another approach could be that for a specific drug, specific IND, what we called, in a clinical trial, you just simply try to convince a regulatory authority that for this specific trial, for this specific drug, you are going to use these specific measures. But this will be only for this specific context. You cannot, let's say, use it somewhere outside for another drug. This was done also this year by the company called Belleferon for their phase 3 clinical trials this year, when they use also accelerometer device, and they look into the time spent in the moderate activity and vigorous activity during the day. It was big change. It was like a first pivotal trial when it was used, right? Problem is the trial failed. Most probably trial failed because of the drug, not because of the measure what they used, but at least it was already shown that it was possible. So this, let's say, two examples of two different approaches, how it could be incorporated into the clinical trial and how it was used. But in general, as I mentioned, so right now it's more about the development of those tools. And because of the development of those tools, so in a lot of clinical trials, like phase 1, phase 2 clinical trial, these tools are incorporated, but they serve maybe like exploratory endpoint primary just to collect the data for the future development. Hope it answers your question.
What's the name of the company? Just like an example that you gave?
Belefron. Yeah. If I pronounce it correctly, yeah. If not, so we can find it together.
If anyone would like to ask more questions, they can find you outside at the stand. You have your
I hope so, yeah.
demo.
So I'm not always going to be close to the stands, yeah, but...
Okay, so last chance for a quick question, if anyone has one. All right.
Thank you so much.
Thank you so much.